Hello.
Hi.
Hi,
Can
how are
you?
you?
Yeah, I'm good.
Good.
I thought if nobody wanted to join you today and well, I don't know. I have a PowerPoint that I've been working on and I could try to explain part of it and see if you think it makes sense.
Yeah, why don't we do that?
You want to do that now?
Yeah, we can do it. We can start off with that. well,
Okay.
well, welcome to the meeting. First of all, let me do a little bit here.
Okay.
Welcome to the meeting. If you're joining us on YouTube, we have a number of things today. I have been communicating with Tharoon and Ujwal and Asmid on their work on the Bacillaria, decomposing the Bacillaria colonies. And they're making some progress, although they're still having some problems with the edges and some of the cases where the cells seem to overlap. I mean, they don't overlap physically, but the algorithm can't seem to pick out the true edge or like an edge that's separable. So they're having a little bit of issue with that, but I think it's coming along pretty well. So they maybe in the next few weeks they'll give a presentation on that. So now, Susan, if you want to present your screen.
Okay, let's see if it'll work. That's the main thing with me. Can I share my screen? Okay, settings, shortcuts. Where would that be on here?
It should be at the bottom. If you go down to the third icon from the left and click
Oh,
on that.
start
Yeah, it's like an
sharing
arrow.
screen.
Yeah.
Okay, so there's share. Um, I'll try to make it work.
Okay.
Yeah. Okay, not happening.
Oh.
All right, then. I don't know. We got buried with a whole bunch of snow, so I don't think they're going to be putting in my fiber optic.
Oh, okay.
So I'm afraid I'm stuck with whatever bandwidth I have.
Yeah.
So it's not working.
Huh. Let me try sharing my screen. See if it works here.
Um,
Yeah, so I guess mine works. Let's see.
okay.
All right, and then let's see. I'll stop sharing mine. Now try sharing yours again.
No, it doesn't even. It's grayed out.
I don't know why that is. Is it the box next to the thought bubble? If you look at the different icons on the bottom.
So there's a share system audio box on the other side, and then on the right side there's a share and cancel thing. And if I push cancel, it works real well.
Oh. Well, okay, well, we can do this another time, I guess.
Okay.
Yeah.
Well, I really wanted to show you, but
I know. Sorry. Yeah, sometimes they have these glitches and they don't, it's hard to resolve them.
It has to do with these experiments that we're supposed to be doing on tissue, and if you push a tissue, is it a trauma to the tissue or is it just a normal movement? Like a normal, I know to build muscle you have to give it micro tears, but so that's sort of an in-between situation, I suppose. But I have a nice graph and try to explain it. Explain this with. Anyway, I wanted to share that.
Yeah. Is it on your home? Is it on your computer, or is it on Google Drive, or what is it? Where is it located?
It's on my computer.
Oh, okay.
I can separate out the slide, and I'll separate out the slide and send it to you.
Okay.
It's a question that needs to be answered before you measure tissue. Are you traumatizing it, or are you pushing it within physiological bounds?
Right. Yeah, that's something actually that I think Yeah, I don't think a lot of the studies look at like when they do a lot of stuff with forces and that, like, and like even with stem cells and that, you have self renewal and you have a lot of, they can repair themselves quite, or they can, sometimes if you stimulate them in certain ways, they can go down, say, a cancer pathway, or they can die, but sometimes they can, you know, basically not be as, they don't, they can regenerate. So, yeah.
One of my big, things that annoy me is, I centrifuged it. You centrifuged it? Like, that's 1,000 times gravity.
Yeah.
The thing's dead. Or I pipetted it.
Yeah.
It might be alive. Maybe. It's like that. Like, people use these laboratory instruments to separate things, and they've killed their specimen.
Yeah. Yeah. Well, yeah. So, like, a lot of times they'll have like different techniques where they spin it at really slow gravity or something, to get it.
Works.
Yeah. There's certain techniques.
Yeah, there are some where they gently rock it or spin it, but no, I was asked to review a paper one time where they were pipetting sperm.
Oh, yeah.
And all I had to say was, this is a really good idea. Someone needs to check out what works and what doesn't. They could get away with pipetting it once, but any more than that, and it had a die-off.
Right. Yeah. Yeah, it happens a lot in experimentation where, some of your sample will die off. It's kind of like you just harvest what you can. You do all these treatments to it, and then a certain amount of the cells will die off, and then you just have to harvest what you can and try to reestablish it. And it's quite it's quite difficult, some of the techniques to. But then there's this additional issue where you say, well, how much am I affecting the cells in terms of their activating a stress pathway versus activating some other thing that you want to activate. So,
and that will be, if you're activating a stress pathway, they'll change their conformation and they'll change their mechanical properties.
Yeah.
So what are you measuring? Apparently you can pre-stress cells. So if you pre-stress the cells before your experiment so you can get nice results, then you're not measuring what was there before.
Right.
And they'll pound ultrasound into something with a whole bunch of clicks, and I'm going, is that a trauma to the tissue? It might be. This might be a trauma if you sent ultrasound, burst ultrasound through something.
Yeah.
So, it's a huge question, actually, for my thesis.
Well, that's good. Yeah. Yeah, it sounds like an interesting question. So I wonder if you could leave the space and then come back in and see if it resets it, because sometimes that works.
Okay. Well, I'll leave and come back.
Okay.
If you want me to.
Yeah. Just try it and see if it'll reset your sharing.
Okay.
Okay. Now we'll wait for Susan to come back. So we have a number of things today. We're going to talk about Google Summer of Code, and we're going to talk about some other things, some papers at the end. So welcome back. Now try sharing your screen.
The share button or the share box is still a gray. It's grayed out.
Yeah.
That's the cancel button isn't, but the share is.
Yeah.
I don't even know how to improve on that. It's probably just my bandwidth.
Yeah, maybe. Okay, well, that's okay. We'll do this another time. Thanks for trying.
Or I can send you maybe the one slide I'm talking about.
Yeah, that'd be good. That'd be good.
Yeah, okay, I'll do that. And and hopefully some other people could have some comments about it, because I am worried about it. I've been asked to use ultrasound as a stimulus for some of my experiments, and it's I don't know which tissues it's a trauma for. So.
Oh, right. Yeah. Yeah. Well, yeah. I don't know if people in our group have a lot of. I mean, Dick might. He has some.
Oh.
Yeah, but I don't know. It's like one of those things. Like, it's. I don't think it's been well studied, so I don't think people have a very hard and fast answer on it. But I think I've seen occasionally a paper where they'll say, like, well, if you do this to the cells, you're going to trigger some other pathway, but then it's just kind of left at that. So
that's a whole area I could be studying with my thesis.
Yeah, I mean, probably not the whole thesis, but maybe oh yeah, by the way, sometimes you can stress the cells or stress the tissues out by doing things to them in the lab. Yeah.
Yes.
Okay. Well, I don't think anyone else is going to show up live here, so I'll go through our agenda here. The first thing I wanted to talk about, actually, last week we talked about the Bacillaria data, which was the data from the book chapter on Bacillaria and the deep learning. There's like a deep learning part and a biomechanics part, and so this is the biomechanics part, and we had these graphs in the paper. So this is what the graph looked like. This was figure 15 in the paper. And so you got these phase diagrams where we had position on the x-axis and velocity on the y-axis. So this is position, or it could be displacement if you rearrange the numbers, and then velocity, which is microns per second. So this is where you have movement in a certain, from position to location, and then this is velocity. As you can see, the velocity increases as you get towards the middle and decreases as you get towards the ends. So that roughly corresponds to each cell kind of like coming to a stop at its extent and then coming back and then making it swing through. And then as it passes the zero mark, it's going at sort of its maximum velocity, and then it does this over again. So it's a nice pattern. And I looked through the data, I went back to the data, and I got some interesting things. First of all, I know that we have a number of different data sets. So this graph actually represents comparing two different cells. So the data was collected on 3 cells in the colony. 3 cells were tracked for their movement. You had so you had 3 different comparisons of 2 cells. So you had cell 1 versus cell 2 versus cell 3, and then cell 1 versus cell 3. So you had 2 comparisons of adjacent cells, and then you had 1 comparison of cells that were separated by a cell. So they were it was down colony effects. So this is the graph for cell 1 versus cell 2. So this is a pairwise comparison where you have the 2 cells that are, expanding up against each other. So they're actually making contact and they're moving against one another. And you can see that this position is sort of a displacement, the way it comes out. So the position is, when it's at this high negative position, the velocity slows down. When it's at this high positive position, it slows down. And then there's when the position is near 0, it's high velocity. Now that's 1 versus 2. This is 2 versus 3. And you can see there's a little bit of difference. Now 1 is at the edge of the colony. So 1 is sort of at the leading edge of the colony. 2 is the next cell down, and 3 is the next cell down from 2. So this is 2 versus 3. So these are 2 cells that are within the colony. So there's no leading edge here. This is within the colony itself. And so you can see there's a little bit different phase diagram pattern. You can see that there's maybe if you call them harmonics, almost like a slowing down, or like, there's some constriction of movement here. So you have like this, again, you go out to -60, you go out to +60, but then you have these slowing down periods at -30, -40, -50. And you also have this kind of noise here, which, this is a noisy pattern. It's not smooth movement necessarily. It's I think it's actually a little bit noisier than this one. So these data are all non-smoothed. So these are just plotted from the raw values. So you can see that there's some noise here in this section, which is where it's kind of making this, it's kind of reducing its velocity. You can see it's gaining velocity here, losing velocity, gaining velocity, losing velocity. And so as it starts to lose velocity, there starts to be some fluctuation in the signal. And this is, of course, 1 versus 2. So this is where the leading edge is moving against an interior cell, and there's a slowing down, there's some instability there. Now in this case, in the comparison of 2 versus 3, you see this, same pattern here where it's slowing down, but also you get this pattern where you get these sort of slowing down events at different positions, positional displacements, or whatever you want to call it. Now, phase diagram 3 then is quite different. This is 1 versus 3, and so this is where you have the leading edge cell and then an interior cell, but this isn't a pairwise comparison. It's a sort of a leading edge versus an interior cell comparison. They're not necessarily coupled in terms of their movement like 1 and 2 are, or 2 and 3 even. They're sort of coupled indirectly. And so you can say this is maybe a second order effect. And so you can see as a second order effect, it's quite different. You have this phase diagram, it's got a lot of noise in it. And it looks like there's a slowing down period where things wind down into this, sort of towards the end of this sequence, it winds down, sort of dampens in this region. So I don't know what this means. This is, you see these different slowing down periods at many scales here, instead of at the extreme ends of the displacement. So you see all sorts of things. So to recap on what these look like, I'm going to use my board here. And so to recap what these colonies look like, you have 1 here, which is this leading edge cell. You have cell 2. So this is 1, this is 2, and this is cell 3, which is inside. And you have other cells out, back this way. And then that completes the colony. So you have other cells behind 3. So it's kind of in the middle. And then 1 is here, and 2 is here, and 3 is here. So you have this 1 versus 2. So you have this leading edge cell versus this cell that's sort of next to it. So they're actually coming into contact, direct contact. And there are all sorts of, they're different molecular sort of like connections here with different types of polymers that both, enable movement in the water column and enable movement against each other. So there's this direct connection. 1 versus 3, on the other hand, are not directly connected. They're intermediately connected through 2. So 2 is moving against 1, 2 is moving against 3, and then you're measuring this 1 versus 3. So this gives you this much different pattern of, where you...
So your 3 is next to the 2, it's not off to the side, or?
Oh no, this 3 is this cell here. So maybe I should label 3 here better. That's 3. So that's your basic pattern. And I mean, I just approximated these positions, but you can see that 3 and 1 are just not connected directly. So that's the big difference there. Okay. So that's the difference in these graphs here. These are the sort of a phase diagram. So you're just plotting 2 variables that are, generate this spherical, or this almost like this attractor basin type pattern. Now, you want to be able to compare also acceleration. So as we know that acceleration is a derivative of velocity. So let's see if I can find a good graph for this. Okay, so this one that's a different one. So this is actually velocity versus acceleration. This is one plot that I'll show. I'll show some other plots. This gives you an idea of like that. If I took that phase diagram and I turned it into a sort of an oscillatory pattern. So I just want to plot out, like, as an oscillation over time, what that phase diagram will look like. And so this is, again, unsmooth data. It's a little rough and, towards the 0 point, but you saw in the phase diagrams that there's, it sort of reaches sort of an end there. There's minimal velocity there. So there's a little bit of a plateau there, but you can see that there's this rough oscillatory pattern. And we have some smooth versions of this in figure 15, which are just kind of like, when we fit it to a sine wave. So it looks more like a sine wave in that, but this is if you don't fit it to a sine wave and just plot out the values. So it's basically this oscillation with a certain period here. Now you see that this blue line here, this is the acceleration. So this is a little bit different. This is where you have this acceleration going on. So yeah, this one is velocity versus acceleration. The velocity, of course, is rough. It's not position, it's velocity. This is the acceleration, the blue line, and you can see that this is not, this has like little peaks in it in different places. So this has a peak, a couple peaks within some of these oscillations. So this cycle here, you have some, some large displacements in terms of acceleration in some of these places, not others. It doesn't look like there's any sort of pattern that jumps out, but this gives you an idea. This is unsmoothed acceleration data. So this is, again, velocity is when it's moving per second, microns per second. The acceleration is microns per second. And you're going to get some, I guess this is in some ways like oscillatory compared to this. It's much, a much shorter period, and it's much more unstable with respect to its own sort of fitting a sine wave. So you can see it's actually, this is something that I actually did do a little bit of signal processing on it. I didn't get any, well, I think I have those graphs in here. But anyways, that's okay. So then there's velocities. This is a histogram here of velocities for cell 1 versus cell 2. So you can see that in terms of velocities, you have, these low velocity moments, and then you have some high velocity moments down here. And you have, like we talked about in last week's meeting, it isn't normally distributed. There's a long tail to this, and you can see that there's it's a fairly thick tail here coming out into this region. So these are higher velocity movements. Okay, so this is velocity versus acceleration plotted out on as a scatter plot. So there's a little bit different way of expressing it. As I showed you before, I had the two traces kind of overlaid. This is where you have velocity versus acceleration here. So the velocity is here, acceleration is on the y-axis, and so you can see that there are high velocity, high acceleration points that are rather rare, and there are a lot of low velocity, low acceleration points. Okay, let's see. We also have, I think these are histograms of accelerations. So this is kind of what I showed you before. This is for acceleration, not velocity. So these are the histograms for accelerations. You see that there's this, there's a very thin tail here. So a lot of cell 1 versus cell 2, the accelerations are, distributed at low accelerations. There are a couple high accelerations in here, but it seems like it's pretty stable if you compare it with cell 1 versus cell 3. There's maybe a little bit longer tail, as you have some events out here, like you have 4 and 5.2. So you have these rare events out here, rare events where the acceleration increases greatly between the two cells. And then cells 2 and cells 3, these are the cells that are interior to the colony. You have the same thing. This is not the same graph. This is a different graph, but you get a very similar behavior. So I don't know if that's what the influence of cell 3, or if it's just the influence of 2 versus 3, and then it's sort of similar with 1 versus 3, but you get this long tail, but these rare events out here where there's a lot of a very few number of high acceleration events and then a lot of low acceleration events. Okay, I think that's all I wanted to show here. I don't think there's anything else. Anyways, I wanted to go over those. I generated a bunch of graphs. Oh, hello, Shruti. How are you? I didn't see you there.
Hey, I am good.
Good.
How are you?
Pretty good. So that's good. I went over those data because we talked about this last week, and I wanted to explore the data. This is pretty rough, this analysis. So I just wanted to give everyone an idea of what that looks like with respect to within the colony, different cells. And we're doing this deep learning exploration of the data. So we're trying to extract numeric data from that. So that might actually give us better data to work with, and maybe some more. We can explore this a bit more.
Have you done other like signal processing where you look at the power spectrum and stuff?
I didn't. I got into it a little bit. I haven't done a lot with it though. I didn't generate any graphs.
Okay. Did you do any more Lisa's U figures of the acceleration versus the velocity, for instance, or?
Yeah, I didn't really play around with that too much. I probably play around with that. Yeah.
Okay. Just curious.
Is that something that looked interesting, or?
Oh, I don't know. I was kind of intrigued about how you can often see things in those circle graphs versus the sine waves. It's just I don't know.
Well, I think, yeah, it's definitely there are different ways you can plot them out. I use the scatter plots because I wanted to see, like, if you compare the two versus just, putting them on top of one another, what are the, obviously they may not share, like, it doesn't show you, like, a correspondence of things in time, but it does give you an idea of, like, are there high accelerations and also, high amounts of velocity. So it's like high velocity versus high acceleration. You'll see like some sort of relationship between them. So that's the relationship that came out. It's not very clear. There's no linear relationship or anything like that. It's just kind of like you get some rare events, and then you see this in the histograms as well, where you get rare event accelerations, and those might be due to, like, things going on as the cells speed up or slow down, so as they hit their limit and then they come back. So
do you think they're on kind of like a spring and bounce back, or?
It may be, yeah. I mean, there's probably this spring-like activity where it goes to the end and then it comes back. And then I know that the connections between the cells are elastic. So there's this elasticity that is, that brings it back, and it doesn't, they don't fly apart, at least not in normal conditions. I think they actually do come apart at some point as the colonies age, but, yeah. So it's very interesting.
Yeah, thanks. Yeah.
So yeah, that's something. So we'll follow up on that in another time. So I want to move on to some other business here. So I know that last year we had Google Summer of Code, and we had a lot of applicants, and there has been a lot of fluctuation in the program over the last 2 or 3 years because of COVID. Actually, it would be the third year coming up this year. So this year what they've done is they've expanded the program. So in the last 2 years, 2020 and 2021, they sort of shortened the time period. So it made it a little bit hard to complete certain projects. And they've had some issues with selecting projects, getting everyone funded and things like that. So this year they're going to, I think, revamp the program. It looks like they put up this blog post a few days ago. And so I wanted to go over this for people and keep this in mind as we're formulating projects for this coming year. So this is for 2022. The way this works is that we usually have solicit ideas for projects now and going up till January 15th, which is when the INCF likes to post these projects and start getting students interested in them. So we have about 2 months to do this. Instead of come up with projects and then write them up as a formal idea, and then make sure it's something that can be done, and then hand it off to our sponsoring organization, and then they can get us spots in the program. So, but this year they're expanding the scope of it. So I like what they've done here. So what they're trying to do here is they're trying to bring in a more diverse group of people with different sort of needs and goals. And so their first thing they've done is expanded eligibility. So they're expanding eligibility now. The only constraint is you need to be 18 years or older. So you don't need to be a university student or recent grad. You don't need to be they've had some technicalities in the past where people maybe who were out of school for a while, or they were in some tenuous position with respect to their enrollment, that was a problem. It's not a problem anymore. So that's good. But the other two things, is that they, first of all, they've modified the size of the project, and that means that they have two different project sizes now. So there's the shorter project, which is 175 hours, which is, basically, I don't know how many weeks that is. It's like 10 to 12 weeks. And then 350-hour projects, which are about 22 weeks. So this is something, I think they've done this in Google Summer of Docs, which is a competing program in the fall. And they have these different program lengths. So the idea would be, if you have a project that can be done in 12 weeks, a small scale project, like say we want to update DevoLearn, again, but we want to do this in a shorter period of time because it's not doesn't require a lot of, lead up development and all this. If we were, creating something new, like if we were creating DevoLearn from scratch, we might want the 22-week project. If we're just updating it, we might want the 12-week project. And then that increases our chances of getting a spot in the program. So this is something to think about. If you want to, if you submit an idea for a project, you want to be a mentor, say, or you want to co-mentor, you can propose a project of these different sizes. And, it allows us to do things like create something from scratch or just, update things. So that's good. Then there's increased flexibility of timing for projects. So related to this, you can do your project, over a 22-week window. So you could do it in more, later in the summer or earlier in the summer. And I've noticed that people have had exams or things like that. You can plan around them. So that's good. And so we have these, I guess they have more information forthcoming on this. My advice on this would be, if you're interested, get in touch now. I can't guarantee you a spot, or I can't guarantee you a project or anything like that. But if you're interested in doing something as a project this summer, let me know. We can maybe create a proposal around that. We can recycle proposals from last year, we can, if Susan wants to pursue the stuff we're doing with axolotls, we can revisit that. I don't know, Shruti, you may have had some things on cellular automata that you were interested in. So, I mean, these are things, again, that we could revisit. And so those are things that we can do in a different
I have
ways.
a question.
Yeah.
Sorry, you can go on. I'm sorry.
Oh, you had a question? What was the question?
Oh, okay. My question was that there are two durations, like 12 weeks and 22 weeks. So that will be project based, like projects will be made in such a way, or it is on the candidate date, like they will decide whether they want to complete the project in 12 weeks or 22 weeks. Like, how is it going to be? Any idea about that?
I think it's like the project can be of the different lengths. So you can have like a long project or a short project. And then you can have like you have flexibility in scheduling. So you could say, I don't want to start this until like August and do a short project and do it to like, I don't know, October, November. Or you could do a 22-week project, and that would give you less flexibility in terms of when you start and finish it, but it would allow you to do a longer project.
Oh, so probably they'll give it like few months, 4 or 5 months, and between that you have to complete your project, and it can be a 12-week or a 22-week project.
Yes.
Something like that.
And then, we'd have to come up with a schedule that would be suitable. So, like it would be like a 12-week schedule, or you'd stretch it out to 22. And then it would have to be like, you couldn't just like take a 12-week project and stretch it out to 22 weeks unless you say, I'm going to take some time off. You have to make sure that it's appropriate for the project length.
For my axolotl images to make a project, well, I've got a new version of the microscope, and hopefully I could get some more images using that system, as well as the older system. Would that help get the project going, or get some more images with?
I think as many images as you can get would definitely be good. And I think another way to, get people interested is, have like and we didn't do this last year, but maybe we could have like a sort of showing some of the microscopes, like just maybe like a video or like some pictures of the microscopes, and just have those like like if people are interested in the project, we can create like an onboarding guide for that. Actually, we had the onboarding guide from last year, and we can just add to that and say, this is this project, and here's what the data collection looks like. Here's what the data look like, and then here's what we want. What we would want you to do, and then people could have a good idea of what to do because they see, what it is. Like this is a axolotl. This is the microscope you're using. This is what the output is. So I think I always struggled sometimes. I mean, people kind of intuitively maybe understand what microscopy images look like, but it's like making the connection between like why you're doing this and then what the goal is at the end.
Maybe a more pictorial representation of what our aim is, and from where we are beginning. So like people, because most of them would be technical people and not having that much background in biology. So I guess a good explanation would be really nice for everyone to understand.
Yeah, I think so.
Yeah, well, it's kind of intriguing because you can say if you follow these cells, they eventually become a brain. So yeah, that's interesting, right there.
Yeah.
So
yeah.
Do a pictorial of it, and maybe a few words in English just so that it's
probably
explained, but not too many words. Just sort of, here it is. This develops like this, and then this is a brain.
I know too many biological words will definitely scare people like us. So I guess there should be a sweet balance there.
Yeah. Yeah, this is called neural tube closure. Yeah, you might not have to go into that.
Right.
Yeah.
But, yeah. Okay. Well, I need to get that put together. I have a new microscope that's easier to work with, and I hopefully near the Christmas time, the salamanders will probably lay eggs. Like I have a friend who lives nearby, and she has a whole group of salamanders, and they tend to lay eggs at Christmas time.
Yeah.
It's what they do.
Yeah.
Especially if they're in a locked lab in the middle of a university in the middle of Christmas holidays. That's what they like to do.
Yeah.
Lay eggs. Especially when you were looking for eggs. Anyways, at least I have usually have access to her eggs. She's very good. She's very interested in my microscope. So between the two of us, maybe we can get some images.
Yeah, that'd be great. Yeah, great. Okay, so yeah, we'll come back to that, and we'll plan out, again, if you have any ideas, if you want to be involved, let me know. So the next thing I'd like to talk about is, I know I've been talking about this for a long time, and I've been putting it off, but I think we're finally here to some extent. This is the DevoLearn technical paper on the DevoLearn platform that I've been talking about finishing up, and this includes my Knox work from this summer. So this is something that I'm still kind of working on getting it organized. I wanted to go through it and revise it because we had submitted it to another venue and they didn't like it, so I wanted to make it larger and then incorporate my Knox work from this last summer. And so this is where this is headed. And so I've been working on this a bit, getting things from my Knox, getting things from, putting in figures and tidying up some of the references and things like that. So again, our authors are Ujwal, Mayok, Mainak, and myself. They have a short abstract, and it's just very short. Just kind of goes over the platform. So this is a summary of why it's important. This is a screenshot from, I think, Mainak's work from this. He made this GIF that we have in here. So this is the accessibility section, which discusses sort of where you can find this, and this is a GUI. So this is all right. So then the statement of need is here. So this is why do we need this, and this kind of goes through the. Again, like I said, this isn't optimal. I need to work on it more, but I think I'm getting some of it in place here, and it's coming along. So. This is a sort of a schematic of how the software works, the GitHub source and the user environment, technical details. So, from my Knox work, we have hyperparameter optimization, meta feature detection. This is both Mayuk and my Knox work. And then the DevoLearn platform, which is the broader platform, which has educational resources and other things. And this is an example. This is the diagram of that. And then future directions, which kind of talk about some of the things we might do in the future or things that need to be clarified. And so then references. So it's still, it still needs a little bit more length, I think, to be an effective preprint. But we're getting there, I think. And again, I want to get this out in the near future here. So maybe before we start in earnest with Summer of Code 2022, because we'll probably maybe have another round of updating to it. So I wanted to get it out by then, and then we can version this as the software develops. So now I'm going to move into papers. We have a couple of submission items. I don't know if we have anything to catch up on, but we have a couple things here. Neuromatch 4.0 is coming in early December 1 and 2. My other group submitted a bunch of stuff, made a number of submissions to this conference, which all got accepted as short talks. So we'll be doing that. I don't know if we'll still be doing any sort of DevoWorm presence at Neuromatch, because it's a pretty abbreviated conference this year. So, but I encourage you to attend this conference. This is the link, Neuromatch.io conference. This is a lot of topics in neuroscience, and they always have some really good talks, some good keynotes, and some good short talks. And then NeuroIPS, which is coming up December 6th through the 14th. So if you're interested in machine learning, deep learning, that sort of thing, NeuroIPS is coming up. So again, if you're a student, you could volunteer or you can attend virtually. You can attend both of these conferences virtually, and it will improve your understanding of these areas. So, the mathematics of DevoWorm, that is something that's in progress. I was going to show it today, but I really haven't made a lot of progress on that to really cover. This is the DevoLearn papers. I just showed that. And then we have a number of different other things that are outstanding. I don't think there's been any progress on those. Last week we talked about a few of these, but I don't think there's anything to talk about on this week. So I'm going to go to papers now. And so this week we have a number of things here. First of all, I wanted to show off these developmental animations. So I like to find these. This one is a bunch of neurons that are migrating into place in development. So you can see that they're kind of migrating around into place. They're making connections with their neighbors. And this is sort of in this time lapse. So you can see this process ongoing. You can see roughly the amount of time it's taking for this to happen. So these neurons are differentiating, forming processes, and then making connections. So this is from 8 hours to, I think, 12 or 13 hours, or actually 0 hours here. So you can see that you have these connections being formed. And as you get past 8 hours, you start to get a lot of activity where they're sort of coming together and making connections. Okay. So then the other thing I wanted to show was this this is just an embryo, and this is an embryo sort of differentiating. This is sort of
looks like a frog.
Yeah, what?
It looks like a frog or a probably Xenopus.
Yeah, Xenopus, probably, yeah. So anyways, this is a nice animation of embryogenesis. So the cells are dividing, and you're starting to get some differentiation here. All right, so that's good. Those are some nice animations. And so then let me talk about some of these other things in here. So this is a paper on graph cellular automata. So this is something that we've kind of talked about a little bit. I think this is the right paper, but I don't know. Okay, I don't know if this is the right one or not. I don't want to cover this one. I thought this was a different paper. It wasn't. I'm going to talk about this one then. This is Turing Morphogenesis. This is a special issue on Turing Morphogenesis, and this just came out recently. This is actually, the date on this is December 27th, 2021. So this is a upcoming issue. It's out now, but it's going to be out in what they call print in a few months. So this is recent progress in open frontiers in Turing's theory morphogenesis. So we've talked about this a lot. This is a picture of Alan Turing. This is his morphogenesis in the background. We've talked about what's going on there, and just a whole bunch of papers on this topic, some of the more recent developments. So one of these things, the first one is you have an introduction to recent progress in open frontiers in Turing's theory of morphogenesis. So this article introduces the special issue. They talk about elucidating pattern-forming processes is an important problem in the physical, chemical, and biological sciences. Turing's contribution, after being initially neglected, eventually catalyzed a huge amount of work from mathematicians, physicists, chemists, and biologists. And this work was aimed towards understanding how steady spatial patterns can emerge from homogeneous chemical mixtures due to the reaction and diffusion of different chemical species. So these species will form in different locations. They will be emitted in different locations. They'll diffuse across the some spatial domain, and then there will be reactions when they meet, and they form things like boundaries, or sometimes they'll form patterns with respect to their spatial distribution. So, while this theory has been developed mathematically and investigated experimentally for over half a century, many questions still remain unsolved. So this theme issue places this theory in a modern context, discussing the current frontiers in a foundational aspect of pattern formation and these reaction diffusion and related systems. So this really kind of goes over a lot of the chemical and biological context to some of these systems. And so there are a number of papers here. There's one where they sort of focus on the spatial dynamics. They focus on the nonlinear systems component of this, and they come up with a general type of bifurcation diagram in a class of reaction diffusion systems, interpolating between qualitatively different kinds of reaction kinetics, which means that you're looking at different types of stability. You're looking at systems that are monostable and bistable, which means they can switch between two states. Their bifurcation results indicate a number of relationships between classical Turing and what they call Hopf bifurcations. So Hopf bifurcations are these bistable systems where you get bifurcations between one state and another, whereas the Turing system doesn't usually have these bistabilities. They usually have this monostability. And then with more exotic structures such as homoclinic snaking, and then you get into this higher level topological set of relationships. So they kind of explore that. Another paper explores, they consider models of bulk and surface reaction diffusion systems, which have become popular models for membrane-bound protein dynamics. And so some of these models are different from our typical reaction diffusion systems in that they describe a different type of pattern formation. This other paper here, Konau et al., provides a comprehensive review of the theoretical and experimental literature on Turing patterns in chemical systems. Unlike the complexity and uncertainty of in vivo systems, chemical systems can exploit extremely well-characterized reaction kinetics and diffusion rates to design and engineer a range of pattern formation scenarios, from the interaction of coupled layers to growing domains using photosensitive reaction kinetics. So you get these different types of, you have this in vivo system that can generate different types of pattern formation. And then we can understand it a little bit better with that kind of a system. And so there are a number of future directions for chemical research and Turing-type patterning. So this is something that we can do with synthetic biology. We can engineer Turing systems that will allow us to look at some of these pattern formation regimes. So this is a nice sort of future directions step. So this is good. They kind of discuss a lot of different aspects of this. So if you're interested in this reaction diffusion model, this special issue is really going to help you sort of think about the possibilities here. Is this the same paper or not? I don't think it is. This is pattern formation from spatially heterogeneous reaction diffusion systems. So this is this paper on spatial heterogeneity and what that looks like. So we know that these Turing patterns are spatially heterogeneous because you form local patterns, and they might repeat, or they might have local differences. So they say in the introduction, since diffusive instability was first shown to result in pattern formation in a model of morphogenesis by Turing, the Turing instability has become a popular mechanism for pattern formation. These resulting Turing patterns have been shown to correspond to various spatial phenomena in chemistry and biology. And so this is a steady state solution of a system of spatially homogeneous reaction diffusion equations. So the classical Turing mechanism involves a single sort of differential equation that allows you to model this process sort of homogeneously. It doesn't matter where you are in space. The same process is working everywhere sort of it with the same type of dynamics, and that's typically how it works. But of course, then the system can lose stability due to spatial perturbation, with the system evolving away from the steady state and into a new state, which is spatially heterogeneous or patterned. So it is possible to consider reaction diffusion systems which are explicitly heterogeneous in space. So what happens here is you have this single equation. It's governing this system, this diffusion reaction system. Then when there's enough spatial heterogeneity, there's a phase transition, and you get this sort of this pattern formation. But you don't actually have equations that describe that process. This just kind of emerges out of the system. So Turing has this unpublished work called Outline of the Development of a Daisy. And so this was something that he worked on as he was developing this Turing instability work. And he didn't really publish obviously didn't publish it. But this, I think, dealt more with spatial heterogeneity. This has been suggested as a factor in the rich variety of patterns seen in nature, including patterns emergent in developmental biology, ecology, and spatial epidemics. So there's been this missing piece of spatial heterogeneity in these systems, and this is what this article is introducing or reviewing to a broader audience. So spatial heterogeneity can be introduced into Turing systems through a variety of mechanisms. You can have space-varying reaction kinetics, which is a popular choice amongst people. You can also have space-varying diffusion parameters, which is also a popular choice. But these aren't things that are, these are just possibilities in your modeling. You still have to capture the spatial heterogeneity, and these models do to some extent, but there are other ways to do it as well. Due to the spatial heterogeneity of these systems, the standard approach for finding Turing instabilities in the parameter set of such systems fails. So the literature concerned with instabilities either considers the case of small spatial heterogeneity, which means it's really restricted to certain regions, or strictly numerical simulations in the case of large spatial heterogeneity. So they haven't really solved the problem of large-scale spatial heterogeneity. We talk a lot about striping, or like the boundaries between stripes. Those are things that are local spatial heterogeneities, but people haven't really come up with good models for global spatial heterogeneity.
This fits in with active matter patterns, because that you've got movement, obviously, when you're creating patterns in a living system. So you have things like vortices, which have always intrigued me. So I really need to get that organized.
Yeah.
Because it's the end of my presentation that I'm doing. So I'm working on that this week. So hopefully I'll get to that point. Because anyways, it all fits together. The Turing patterns, along with the active matter, will create patterns in living systems.
Mm-hmm. Yeah, I think that's good. And I don't know. I haven't really gone through this special issue too much, but I know that there are probably a lot of articles there that might help make that bridge. And I don't know if people are making it explicitly in terms of like some of the soft active materials literature, like if they're bringing that in explicitly. But I think, there are a lot of links there.
Well, active matter is definitely a topic in physics.
Yeah.
Like physicists are the people who are studying this. So,
yeah. So, but also thank you, Shruti, for attending. I see Shruti had to leave early. So let me go on to another paper or two. So this is the Turing Morphogenesis special issue. Also, I have this paper that Dick brought up here on tissue biomechanics. This is something he sent out. So this is tissue biomechanics during cranial neural tube closure. measured by Brilliant microscopy and optical coherence tomography. So this is something that Susan is probably very familiar with to some extent. So this is the background of this paper is embryonic development involves the interplay of driving forces that shape the tissue and mechanical resistance that the tissues offer in response. While increasing evidence has suggested the crucial role of physical mechanisms in embryo development, tissue biomechanics is not well understood because of the lack of techniques that we can use to quantify the stiffness of the tissue. So they use two all-optical techniques, optical coherence tomography and Brilliant microscopy, to map the longitudinal modulus of the tissue. And this is in mouse embryos. They're able to get a look at the neural tube region at embryonic day E8.5, which is fairly early in mouse development, and E9.5. So these are two data points with submicron spatial resolution. We found the modulus of tissue very distinctly within the neural tube region of the same embryo, and between embryos at different developmental stages, suggesting our technique has enough sensitivity and spatial resolution to monitor the tissue mechanics. And this is a non-invasive technique, so they're able to do this. Let's see if they have any images in the paper. So let's see, let me zoom in here. And so this is a representative 3D images of mouse embryos acquired by OCT, which is this optical coherent tomography. This is E8.5 here in A, and this is E9.5. So you can see that here's the neural tube up here, and you can see that there's development here from E8.5 to E9.5. So this is right when the neural tube is sort of forming. So this is fairly early in mouse embryogenesis before you really get this is going to become the brain. So this is going to become the central nervous system, but you need to have this neural tube, and the neural tube closes, and then you get this structure that will then become the brain. So we have this, so we can see this difference here, and this is a nice image because it does show sort of the difference here in these 2 points. So let's see. The modulus of neural tube tissue is a gradient along dorsal ventral direction, and fusion region is much softer after closure. So they can actually measure now the hardness or softness relative to some metric of this neural tube. And so we know that like there are different parts of the neural tube, and there's a hardness and softness of those tissues depending on their morphogenesis, as they're closing or as they're closed. There's also a gradient in a dorsal ventral direction. So there's, they report these results here. Then they have this ectoderm layer, which can be distinguished from the neural tube. So you have this ectoderm and is much softer. So this technique will not only measure the hardness or softness of the neural tube at different points in development, but can also get the ectoderm layer distinguished from the neural tube itself. So you get these differentiating layers, and you can actually look at that. So the process of neural tube fusion and closure involves several critically important cell activities, such as apical constriction, convergent extension, and cell migration. So these are all things that involve sort of thickening, lengthening, elevating, and bending this neural tube tissue. So the tissue itself is changing shape, it's changing its consistency, and then this is all something that we can measure in this using these techniques. So this is, this opens up, I think, a lot of avenues for some of the physical, sort of characterizing some of the physical aspects of these processes. Did you have anything to say about this, Susan?
Oh, they should definitely try axolotl. I can think of an axolotl model for some of the migrating cells. There's the white salamanders, like they call leucistic. They're apparently some of the melanin cells are blocked from migrating, so they're white, not because the they're naturally that way, but somehow they get blocked in that phenotype. I don't know. It's too bad they couldn't try this on axolotl, because there's a number of interesting things that I know go on in axolotl development that would be interesting to see.
Right.
Yeah. So.
Yeah, this is a nice paper. I think they just, this is a proof of concept. Of course, the proof of concept could be maybe more interesting, but I think they've got a good. So this is like showing some of this, part of like different parts of the neural fold, and like showing the softness and stiffness peaks. So there are these stiffness peaks. The fusion region can be defined here as sort of a dip in the stiffness. It's a relative soft region, and then you can see the regions around it. So this is the segments that they divided into.
If I had a Brilliant microscope, I could do this, because there's the lab I'm working with that has the OCP, and then you need the Brilliant microscope to measure some of the rest of it. Brilliant microscopes are very sensitive to water content, but they can definitely, give you an idea about the stiffness of the tissue or the elasticity.
Yeah.
Anyways, this is a nice article.
Yeah. So this is, I don't know, there are a lot of images in here. And then that's the end of the paper. So yeah, this is a good paper. And then finally, I'd like to end with this paper on Wired for Insight. This is a C. elegans paper on C. elegans. I think it's on the, let's see. So Wired for Insight, recent advances in C. elegans neural circuits. So this is just on neural circuits. And again, we have some really nicely worked out neural circuits in C. elegans for behavior. So this is kind of a review of this, kind of goes through some Commectomics advances. So this is good if you want to know the state of the art in the field of neural circuits in C. elegans. So this is, the abstract says, the completion of C. elegans Connectomics 4 decades ago. So C. elegans Connectomics has a head start on a lot of other organisms. This is where the entire connectome had been worked out. And, we're now kind of figuring out like some of the details in terms of development, in terms of, behavioral connectivity and plasticity and things like that. But the connectome for C. elegans has been worked out, and it's been a long time that it's been worked out. So this is a long guided mechanistic investigation of neuronal circuits. So we've known the connections, but we've not known necessarily how they work in terms of behavior. And so these are things that OpenWorm is deeply involved with, trying to figure out like how these circuits work, given some, given behavioral data, given neural data that we have, and just kind of working that out. Recent technological advances in microscopy and computation programs have aided re-examination of this connectome, expanding your knowledge by both uncovering previously unreported synaptic connections and models for neural networks' underlying behaviors. So there are also a lot of these synaptic connections, which have different, strengths, have different, sometimes they're active in some parts of life history, like in development. Sometimes they change in adulthood. They can change with aging. They can change with behavior. So they're in, you have like the Dower stage of C. elegans development where, there are significant changes to the nervous system in terms of synapses. So these are all very important things. Combining molecular information from single-cell transcriptomes with elegant tools for cell-specific manipulation has further enhanced the ability to precisely investigate individual neurons. So now we can get single-cell measurements of transcriptomes. We can get information about what genes are expressed in single cells, one cell versus another cell. So this is very useful information. And this really, in C. elegans, it's really nice because C. elegans is relatively invariant with respect to these things. So, in other organisms, it would be very hard to put together all this into a single, model of the brain or a model of a neural circuit. So we're at an advantage of C. elegans. This mini review aims to provide an overview of new information on connectomics and progress toward the molecular atlas of C. elegans' nervous system, and discuss emerging findings on neural circuits. So this kind of goes over some of the advances in connectomics. It started with Sidney Brenner, and it goes to John White, who did a lot of the early work on this, publishing a reconstruction of the entire nervous system back in 1986. So these are, there are all sorts of different reconstruction techniques that you can use. These are aiding the expansion of connectomic studies to other organisms. They use a lot of electron microscopy to do this type of work. And we also have the Cook paper that is fairly recent. This reanalyzed the original electron micrographs of pharyngeal nervous system neurons and added anatomical weights for synaptic strength. So, we're trying to infer different synaptic strengths from the data. This reevaluation of the pharyngeal nervous system connectome not only revealed previously unreported synaptic connections and additional details of extensive cross-connectivity, but also provided evidence that the pharyngeal neurons may have both have sensory and motor characteristics. So in C. elegans, you have these sensory motor neurons that are both sensory and motor. And so this is a table of the different experimental technologies for extracting these connectomes. So, you have electron microscopy, you have genomics, in which you can look at what genes are doing in these connectomes. You have TRAP-seq, which is a technique for protein expression, looking at protein expression. You have ribosome-tethered GCAMP, which is another technique, and then you have chemo and optogenetics. So this is where you actually put fluorescent proteins into the organism, and you use a certain frequency of light to excite those fluorescent components, and you can see this in the active neurons. So this is a very powerful technique. We also have sexual dimorphism, C. elegans. You can look at the differences between the male and the hermaphrodite connectome. This has been some more recent work on this. There was a paper a couple years ago where they mapped out the entire connectome of the male, which is a little bit different than the hermaphrodite. We also have new data on the development on early adult and L4 stage C. elegans, looking at the connectivity as well as the number of cells that are there. We have finer structures and expanded microdomains. So we have these super resolution microscopy techniques that allow us to look at some of the microstructure of the cells, looking at some of the things going on around the cells. We also have this molecular and cellular connectomics, where people are investigating different aspects of gene expression and protein expression and things like that. You can also do genetic manipulations, of course, which is what we mentioned with the GCAMP technique, and also NeuroPal, which is a neuronal polychromatic atlas of landmarks. This allows us to identify things, identify different types of neurons, which can be distinguished by their gene expression. This has been an open topic that, we've been trying to figure out how many cells there are in the human brain, or how many cells are even in the C. elegans brain. We assume we know because we have them labeled through, anatomical observations. But of course, if we look at the gene expression profiles, we find out that there's potentially a lot of different types and subtypes of cells that we really can't identify using traditional microscopy techniques. So these are things that we want to know as well. So yeah, there's a lot of stuff in here. There's they're talking about building a single cell molecular expression atlas. This is something that is based on early transcriptomic analysis of embryos and L2 larvae. This is something that it would help understand the different developmental times of cells from gastrulation to terminal differentiation. and then building this. This is Vizcello, which actually exists as a database. I don't know if we've had this in a previous meeting, but this is a really nice database that has a lot of different cells and C. elegans at different points in development, and it actually will allow you to visualize this using some of our multidimensional techniques, visualization techniques. And so that's a nice tool that people have developed. There's also this CNGEN initiative, which is where they want to create a complete gene expression atlas for each neuron from larvae to adult. And so this uses bulk RNA-seq, which is this technology where they can sequence different transcripts and look at their prevalence in different cells. So you're looking at RNA transcripts, and you can actually identify, maybe, different transcripts have different levels of expression in different cells and identify differences between cells in that way. It's a very difficult technique to sort of get to definitively say whether you have one cell type versus another. Usually the levels of RNAs are fluctuating so much that you can maybe get to, you can maybe uncover unique splice variants or other types of things, but it's a little bit harder to understand than looking at cells under a microscope. So they kind of go through a lot of this, and then advances in neural circuits. So, we have a lot of different things that we can do with neural circuits. There are a lot of, we've kind of got nailed down the basic neural circuits, but we have yet to really understand the roles of local interneurons and some of these other things. We also don't know exactly the extent of neuroplasticity that exists in C. elegans. We assume there's very little, but they may or may not be true. There are ways you can manipulate neural circuits using ablation techniques. So you have that. You also have other techniques where you can modulate food supply, or you can modulate things like dopamine and neuropeptides, and you can get, different modes of locomotion, which actually allow you to tease out some of the circuit. So here's the connectome here. In the adult, you have the nerve ring, the head ganglia, the ventral nerve cord, motor neurons downstream here. This is the dorsal nerve cord, which goes up the top of the worm and goes from head to tail. And then you have these tail ganglia cells, which is, this emerges fairly early as well. And this is like the tail end of the nervous system as well. So this is all one connectome here. And it does, these cells participate in different behaviors, chemosensation, motor behavior, other types of things. And these are the maps that we usually use to look at it. So we have these different cells. These are the names of the cells, but in terms of the nomenclature, and then you propose these wiring, either an inhibitory relationship or an excitatory relationship. So one cell either turns another cell on or turns another cell off, and then this is how this works. So given the stimulus, or given the goal, of moving left or moving right, these neural circuits behave in different ways. And so that's about it. And you get like circuit regulation beyond neurons, even with the neural gut axis. So they even consider, outside of the traditional connectome, what other parts of the body you're doing. And in C. elegans, since it has 900 or so cells, you can do this quite easily and understand some of these relationships on a cellular level. And so this is another example of bacterial aversion and how the motor circuit responds to this. So this is something where if they're eating bacteria, or if they, if they ingest bad bacteria or something, they have an aversion to it, and they actually reverse away from the stimulus. And this is a signal that they get from their gut. They get intestinal bloating. And then, this is like if you eat a bad piece of cheese, what do? You don't eat the cheese anymore because you get the signal from your gut telling you not to do it. So this is the nice C. elegans model of this sort of aversion to a stimulus. So that's all I'm going to say about that. I think we'll end for today. Thank you for attending. Did you have anything to say about that paper, Susan?
Well, it looks interesting. I'll probably try to read it. And I know the gut-brain axis is important for mammals as well. So for humans, actually. So it's an important, well, a study. And if you can study it in C. elegans, you might get some insights. So
Yeah.
hopefully they can figure some things out because, anyways,
Yeah, it's a nice stepping stone to other connectomes.
yes. So thanks for the papers. I managed to capture them. I got a new keyboard, one that does screen capture, so
Okay.
I was able to capture the titles of all of those papers.
Oh, good. Good.
Yeah.
And I'll send out. I can send them out on the Slack. I could send them to you via email as well. So.
Okay. Well, I do have them, and I do have that one paper on development. I gave it to Dick, and he gave it to you, and I gave it to Dick. We've been seeing that one on embryogenesis in the news because it's a recent paper.
Okay.
So
Yeah.
yeah, and goes right along with some of my research.
Yeah.
I'm still at a more basic point with my research and doubt if I'll be doing mouse embryos.
Yeah.
But it would be more of a postdoc thing, and I haven't even got my thesis organized, so it's a bit of a ways down the road.
Yeah. Sounds good. Okay. Have a good week.